and welcome back everyone to NewtonGATE, where we make your dreams come true. Today, I'll be discussing a new topic of class 12 biology, reproduction in flowering plants. So, as we know that flowering plants consist of four typical parts, that is the calyx, corolla, androecium and the gynoecium. Okay. Now, androecium and gynoecium are the male and female reproductive parts of a flower. Okay. And petal and sepal consist of, petal is the unit of corolla and calyx is the part which consists of sepals. So, sepal are the units of calyx. Okay. So, in the flower, the male and female reproductive structures, the androecium and gynoecium, differentiate and develop. The androecium consists of a whorl of stamens representing the male reproductive organ, whereas the gynoecium represents the female reproductive organ. Now, we will discuss about the structure of stamen. Okay. Now, typically, the stamen consists of anther, which has got two parts. Okay. One is the anther lobe. Okay. And this one is the slender or stalk filament-like structure, or termed as anther filament. And there is a connective which connects the anther with the two anthers. Okay. So, typically these are the part of a anther. A typical angiosperm anther is always bilobed in nature, with each lobe having two theca. That is why they are termed as dithecous. Okay. As because they have two theca, they are termed as dithecous. The anther is a four-sided, or we know that tetragonal structure, okay, consisting of four microsporangia located at the corners, two in each lobe. Means one anther has got two lobes and each lobe consists of two microsporangia. Okay, so that there are four microsporangiums are there. The microsporangia develop further and they become pollen sacs. So, this is the transverse section of a typical microsporangium. Okay, you can see here that it is almost circular in outline. Okay, it is circular in outline and it is generally bounded by four wall layers. It has got four wall layers. You can see here that this is the outermost one is the epidermal layer, that is the epidermis. The next comes the layer that is endothecium layer. Okay. Then the third layer is the middle layer and the fourth one is the tapetum or the innermost layer of the microsporangium. Okay. So these are basically the four parts, four wall layers of a typical microsporangium. Now, what is this part? What is this part? Okay, this is nothing but this is a group of compactly arranged homogeneous cells. Okay, homogeneous cells arranged in the center of the microsporangium, in the center of each microsporangium. Okay, there is a sporogenous mass of cells. This is also a sporogenous tissue. So there are four microsporangium and then there are compactly arranged sporogenous tissues, that is 4. 1, 2, 3, 4. Okay. So this is the transverse section of a typical microsporangium, you can see here. Okay. Now we have studied that there are four layers. Now what is the function of these layers? Okay. Now the three layers, that is the epidermis, endothecium and middle layer, helps in protection and help in the dehiscence of anther to release the pollen grains. Okay. Now, and the innermost wall layer is the tapetum. Okay, that nourishes the developing pollen grains. So these are the functions of the wall layers present in a microsporangium. Okay. Now we will discuss what is microsporogenesis. So microsporogenesis is basically the process of production of microspores from a pollen mother cell, that is PMC, is termed as microsporogenesis. Okay. This is the microsporogenesis process. Okay, this is the flowchart diagram. So here we will discuss about the process in detail. What happens when anther develops? Cells of sporogenous tissue undergoes meiotic division, okay, to form microspore tetrad. So this is the microsporogenous tissue that undergoes meiotic division to form this microspore tetrad. Okay. So sporogenous tissue in microsporangium acts as they are responsible for producing this microspore or pollen mother cell that undergoes meiosis and forms this microspore tetrad. Okay. So each cell of sporogenous tissue is capable of giving rise to a microspore tetrad. Microspores, as they are formed, are arranged in a cluster of cells known as microspore tetrads. As anthers mature and dehydrate, the microspores dissociate from each other and matures into pollen grains. So this is the schematic diagram of microsporogenesis. Okay, these are the step by step of microsporogenesis process. Okay. Now, pollen grains, we have seen that pollen grains have been formed in microsporogenesis as the last product. Okay. Now, what is pollen grain? Pollen grains represent the male gametophyte in different species of flowering plants. They are generally spherical. You can see here they are generally spherical in nature and has got two prominent wall layers. Okay, one is the hard outer layer called the XII and one is the inner layer. Okay, XII is made up of a chemical known as SPOROPOLLENIN. Okay, which is the most resistant organic material known. What is the function of SPOROPOLLENIN? Now, SPOROPOLLENIN can withstand high temperature, strong acids and alkali. No enzymes that degrade SPOROPOLLENIN is so far known. Okay, so this is basically the outer layer I am talking about. Okay. Pollen grain has prominent apertures called germ pores where SPOROPOLLENIN is present. They have got some germ spores where SPOROPOLLENIN is present. Now, pollen grains are well preserved as fossils because of this particular SPOROPOLLENIN. Okay. It also exhibits a fascinating array of patterns and designs. Now, the inner wall of the pollen grain is called the intine. Okay, it is a thin and continuous layer made up of cellulose and pectin. And the cytoplasm of pollen grain is always surrounded by plasma membrane. This is the pollen grain structure. Okay, this is the sculpturing. XII is the resistant outer wall that is composed of SPOROPOLLENIN and which is derived from tapetum. Tapetum we have already discussed in the last part, that is the microsporangium wall layer. The innermost wall layer of microsporangium is tapetum. Okay, so it is present in spore walls of all plants. Now, intine is the inner wall that is composed of cellulose and pectin. Okay, so this is the pollen grain structure. Now, development of pollen grain. Okay, so as we see here, there is a pollen grain structure which has got generative cell and vegetative cell, that is vegetative nucleus, and there is a germ core also. So what happens when pollen grain matures? It has got two parts or two cells, like the vegetative cell and the generative cell. The vegetative cell, as you can see, is bigger, has abundant food reserve and large irregularly shaped nucleus. Okay. Whereas the generative cell is small and floats in the cytoplasm and the nucleus. In most of the angiosperms, like 60% of angiosperms, pollen grains are shared at this 2-cell stage. And in the remaining species, the generative cell divides mitotically to give rise to the male gametes before pollen grains are shed, that is 3-cell stage. So basically we can see that in maximum cases pollen grains are shed at 2-cell stage, and in few cases pollen grains are shed at 3-cell stage. Okay. So in the last section I have given some important questions. Okay. These are very important for your exams. So please practice this at home. Thank you NewtonGATE Jams for watching our video. Like, share and subscribe. Follow us on Facebook, Twitter, Instagram. For more details, visit our website www.newtongate.in, or you can call us directly on the displayed number. Thank you.